In this video we will look at the different steps that we need to take in order to prepare our biological sample for electron microscopy. Now, the information in this video is by no means meant as a detailed guide to the ins and outs of each technique. Sample preparation can be very complicated and it's a science in itself in order to do this right and to do it with minimizing the chance for artifacts. But what I want you to know from this video is to understand the different steps in broad lines, how they can be performed and what the considerations are for using these techniques. Now, the first step is the fixation. In a broad sense, there's two ways in which we can fix our sample. The first is with chemicals. These are, for instance, aldehydes, or we can use osmium tetroxide, which, as we see later, will also act as a stain, so as an electron contrast agent for our microscopy. And the other way of fixing is by cryofixation. So in cryofixation, we rapidly freeze the sample, but the important thing here is that we should freeze the sample in an amorphous fashion in order to preserve its nature structure. So we should realize that if we give the water time to freeze and form ice, this will distort the sample because ice expands, and also it dislocates the proteins and brings it out of the ice crystal. So in order to rapidly freeze, we can use two approaches. The one is plunge freezing, where we have a very small drop of material that we shoot into a bath of liquid nitrogen. And the other is to first apply a high pressure and then do a high pressure freezing in a dedicated instrument. In the lectures about cryo-electron microscopy, Arjen Jacobi will talk more about these cryogenic procedures. After the fixation, the sample is ready to be sectioned. So this can be either in cryo conditions or after embedding the material in a polymer resin so that it becomes amenable to sectioning. Sectioning typically is done with a microtome, either with a glass knife or in an ultramicrotome with a diamond knife. You see a few examples of a microtome in the images here. The images to the right illustrate the basic operation of a microtome. So the block of material embedded in a polymer resin is attached to a holder, and you see below this holder a small water bath located, and the edge of this water bath facing the holder and the resin block contains a small glass or diamond knife. Now the holder brings the block over the knife, cutting off a thin slice, and this slice now falls into the water bath, keeps floating and ready to be picked up. By manipulating the holder and the sample over and over the knife, we cut serial sections which can all be picked up from the water bath. By adjusting the translation of the holder, we can tune the size at which the knife cuts the sections. We've seen later that typically this is from 50 to 200 nanometers. So after sectioning, the sample is ready for an additional staining. Now, an important stain is osmium. Osmium is also used as a fixative, so it may already be present in the sample. And basically, osmium metallizes the membranes in the material, so if it gives contrast on the membranes and thus identifies the different biological features that we have in there. But we can also further stain the material after the steps of embedding and sectioning, which is what we call a post-embedding or a post-sectioning staining. Typically, we use a metal salt like uranyl or lead for this purpose. Now you see an example of a biological tissue section heavily stained in the images here. Note that the left image has a scale bar of 2 micron, and in the upper left corner we see a cell nucleus. Note also the wealth of structural detail that is visible in this image. The right image with the red box surrounding it shows a detail of the red boxed area in the right image, which further illustrates the amount of high resolution detail that is apparent in these images. Now, besides applying these general stains, like the osmium, the uranyl, or the lead, we can also label specific proteins or biological molecules in the material, for instance with immunochemistry. Of course, we have to now realize that we apply the immunochemistry either before going into the process of embedding and sectioning, when there's a lot of epitope available to which the antibodies can bind. But we can also do this after this whole procedure, but then the sample may be slightly etched in order to remove the upper layer of polymer. And of course, in this case, we may be limited because some of the epitope may have been lost by the action of the polymers and the stains on the biological materials itself. In the image here, you see an example where 2 nanoparticle labels have been added. I will zoom in a bit, where you can see gold nanoparticles located mostly in the cell nucleus. These are 50 nanometer particles. And you also see quantum dots, which are located in this case on the insulin granules in this red pancreas section. You see the quantum dots are slightly smaller, they are slightly less dark compared to the gold particles, and they are more densely located on the insulin granules than the nanoparticle label is. Now we know that in light microscopy, one of the big advancements of the last decade was that we have the possibility of having genetic fluorophores. So fluorescent molecules, labels, that can be genetically expressed inside an animal or a cell. And of course, also for electron microscopy, people have been working in this direction. And the first genetic label for electron microscopy was the so-called mini-SOC that you see depicted here. So how this works is that it's a fluorescent molecule where exciting the fluorescence leads to the generation of reactive singlet oxygen, which further can be used to oxidize a substance that we can add later called diaminobenzidine. And this diaminobenzidine can subsequently be polymerized, leading to black deposits on the places where the mini-SOC was expressed. These black deposits are similar to what we would see with osmium tetroxide, only now they are very much localized at the positions where this label was expressed and where we have actually carried out the fluorescence excitation. So you see an example in the image here. This is from the original paper. You've seen first on the left, you see the images in fluorescence, and now if we look at the zoomed in images, we indeed see the dark black deposits on the positions where we also have the fluorescence expression. Now in the past few years there have been more optimizations to these probes, and actually there have also been probes developed that for the oxidation do not rely on fluorescence excitation, but actually use chemicals like horseradish peroxidase in order to carry out this oxidation. This is a field that is still very much in development at present. Now we have seen all these different steps, and all these different steps can be conducted in many different ways. I already expressed at the beginning of this video that the sample preparation for electron microscopy is a science in itself. You see here a very broad depiction of different electron microscopy steps and how they can be connected in a full preparation scheme. But the actual schemes can be much more complicated and much more versatile than depicted here. There's many variations, many alternative routes. We can cryofix a sample and bring it later to room temperature. We can also start with room temperature fixation, bring it to cryo for dissectioning, and then go back to room temperature again. A lot of different possibilities. Typically, a sample preparation protocol is optimized for a specific cell type, for a specific investigation, for different labels, or for different staining procedures. Now, electron microscopy sample preparation is known for the possibility of artifacts occurring. But we have seen before that an electron microscope also allows us to see all the structural detail that is present in a sample. And actually, this also allows people to see the artifacts that occur in a sample. So you see that in the images shown here, where the left column of images shows images fixed with 2% paraformaldehyde, and the right image shows a higher concentration of 4% of this fixative being used. And now in the rows you see the effects of the different permeabilizing agents that can be used. Methanol, Triton is all mentioned in the caption. So we see, if we compare these images, that artifacts may occur, holes, ultrastructure may be less preserved from one image to the other. So in general, electron microscopy sample preparation protocols are very well validated and optimized to preserve the native state of the biological specimen for as good as possible. So this brings me to the end of this video, but not before I have recapped the main concepts of this lecture. So first, we started out by looking at the signals that we can detect in a secondary electron microscope, and you should be able to tell where in the sample these originate from. These are the secondary electrons, which mostly give topography contrast, and the backscatter electrons, which mostly give us contrast based on atomic number, nucleus size. So you have to know the difference between secondary and backscatter electron detectors and how they operate. You have to understand how a sample can get charged and what the different regimes for charging are. Finally, in the second part of today's lectures, we looked at sample preparation for EM, where we looked at whole cell, or actually macroscopic material visualization versus thin section imaging. We've seen that there are different steps in the sample preparation for electron microscopy: fixation, dehydration, embedding, sectioning, and staining, or adding labels for electron microscopy. So you have to understand why these different steps are applied and what the considerations are in applying each of these steps. We've looked briefly at cryogenic versus chemical fixation. You should know the pros and cons of both. And we've looked at the sample preparation diagrams or workflows, how they could relate these different steps with respect to each other.